Hello students, here I am to do the most important animal of unit 1, and the topic in which it is included is life history of sessile and pelagic tunicates, Anasidian, Pyrosoma, Salpa, Dolulum and Oikopleura. As you know, we have already done the 4 animals: Pyrosoma, Salpa, Dolulum and Oikopleura, and here I am to proceed with the 5th animal, which is an Ascidian, and we will take Herdmania for representing an Ascidian. So let's do the life history of Herdmania, part 1. Now, what is Herdmania? Herdmania is a sea animal found in the sea, and the fishermen who have a little knowledge of zoology and they look at animals as they appear to them, they have kept the name of this animal Undapasi, and as you can see in the text, Unda means round and Pasi means weed. The kind of round weed, they think it to be a round weed like structure. And the second name is Mulai Kana. Mulai means breast and Kana means teats. So they are, because it's a bag like structure and it has 2 siphons which look like teats, so they call it the breast with 2 teats, and in their language it is called Mulai Kana. The systematic position, if as a zoologist we go and look at the animal, then we have to know the systematic position and all its detailed features according to biology or zoology. So first and foremost, let's do the systematic position. In the systematic position, the animal comes in phylum Chordata, and its subphylum is Urochordata, also known as Tunicata, having a tunic. The animals are covered by a tunic, and the class is Acidiaceae, order being Acides simplicis, and the genus is Herdmania, and the species we're going to do is Herdmania pallida. Now there are all other species also, there are some common species like Herdmania celonica, Herdmania mauritiana, Herdmania inurensis, but the detailed description in this lecture will be of Herdmania pallida. One more thing which I missed is the class we're doing is Acidiaceae, and as we've done the other 4 animals, they are being included in the classes Larvaceae and Thaliacea. The animal Oikopleura was in the class Larvaceae, and the other animals Salpa, Dolulum and Pyrosoma, they all belong to class Thaliacea, and this is the 3rd class Acidiaceae. So this is how we are doing all the protochordates, representing each of its class by different animals, and the maximum number of animals we've done is from class Thaliacea. So here we are doing class Acidiaceae, and the animal included in it is Herdmania pallida. Now next comes the history. Before 1888, Herdmania was known by name Rhabdocynthia. It was only in 1881 that Lahill proposed the name Herdmania, and other scientist Herdman, after which the name of the animal goes. In 1906, he gave a brief account of the anatomy of Rhabdocynthia. So this was about the history. Now we come to the habits and habitat. And as I told you, it is found in sea, so it is a marine animal which is found at all depths. Like it can be found in shallow waters, it can be found in deep waters. So it is, in that sense, it is kind of present on all levels of depths in the sea. It is solitary and is found attached to some substratum by its foot. It is solitary, but sometimes it is also found in groups. When it is detached from substratum, the foot acts as a balancing organ. It usually exhibits the phenomenon of commensalism. Commensalism means it lives in association with some other animal, and the association is beneficial to both of the animals. To Herdmania also, on which it is dwelling, on the animal which is it is dwelling, it is beneficial to that animal also. During this process, during the process of commensalism, Herdmania offers protection to the mollusk from its enemies, while in turn mollusk provides more food, oxygen, and a large area to disseminate its progeny. The test of Herdmania is inhabited by several organisms. Green seaweeds commonly grow on the test and surround the animal completely. Hence you remember the name Undapasi. It may be due to this, because the animal looks like a weed, as the weed covers it completely. The common animals which are usually found attached to the test are lamellibranchs, sea anemones, mollusks, barnacles, gastropods, some hydroids, etc. Female specimens of certain decapod crustaceans have also been recovered from branchial and atrial siphons, and that place is very good for them because the water going in and out can carry their waste material and their reproductive structures. Due to the flow of water, all these things, they remain in a flow, which is good for the animals. Herdmania feeds upon diatoms, algae, and infusorians. A constant current of water enters the body through branchial aperture. The constant water, it enters through this branchial aperture, and after circulating through the body, it goes like this, and then here there is the stomach and intestine, and then the rectum, cloaca, and it opens in the atrial siphon. The current of water helps in all the activities of the animal, like respiration, excretion, nutrition, reproduction. So this was all about habit and habitat of the animal. Now the distribution. Distribution means the occurrence of the animal on the global map. Herdmania is almost cosmopolitan in distribution and is found in all the seas except Arctic. Though there are about 16 species reported from different seas of the world, yet there are only 4 species reported from Indian Ocean. And the names of these species I've already taken, but let me repeat. The species occurring in Indian Ocean are Herdmania pallida, Herdmania celonica, Herdmania mauritiana, and Herdmania inurensis. This was kind of introduction of, or detailed introduction of Herdmania. Now let's go to the another. Now we are going to do the structure of the animal. You must be mesmerized till now that what is the structure, the kind of structure you are looking at. It at all does not seem at all like a chordate. So if for your knowledge, let me tell you, the chordate characteristics are all shown by its larva, and then the larva, it goes through retrogressive metamorphosis and the animal becomes degenerate, and it becomes sessile. After a free moving larva, the animal becomes sessile. So these are all marvels of evolution, and these things happen. And now all these detailed explanation of this will come in the reproduction when you do the structure of tadpole larva. Its larva is known as tadpole, and the retrogressive metamorphosis to result in such a degenerate animal. So let's first do, this is the animal Herdmania, as you can see, and we will go about its structure topic wise. The first is shape and size. As you can see, it is oblong in shape. The upper part is broad, and the lower part, which is known as the foot, upper part is the body and the lower part is the foot. The body is bag-like and broad, and the lower foot is a kind of triangular structure pointing below, and this part is pointing so that it can be embedded in the sediment or in the sand wherever it is found, and so it becomes a little pointed and it remains embedded inside. The only structure visible is the body. When this animal, as we had seen in commensalism, it just attaches to the shells of the animals, so the shell is a very hard structure. In that case, there is a very less development of the foot, and the body, it becomes concave from below, from this area it becomes concave, and it attaches to the shell of its commensal, whatever the animal is. So due to this shape, broad anteriorly and narrow posteriorly, it is called it is in the shape of a toy balloon. And as you can see in the body, there are two apertures, the branchial aperture and the atrial aperture. The anterior end, here you can see this is the anterior end. The anterior end is marked by the branchial aperture, and I will come to the symmetry later. The dorsal part is marked by the atrial aperture, and the atrial aperture, it is directed upwards, and the branchial aperture, it is directed sideways. At the distal end of the of the siphons, this is the whole thing, this whole thing is known as the branchial siphon, and the distal end of it, the extreme end of it is perforated by apertures. In the case of branchial siphon, there is a branchial aperture present, and in case of atrial siphon, there is an atrial aperture present. These are the openings. This is the entering point of the water, and this is the exit point of the water. Now in this branchial siphon, there are many kind of muscles present, mainly two kind of muscles present. These kind of muscles, in the lecture more diagrams will come and you will be able to see them clearly. They are annular muscles which are extended like this, and there are longitudinal muscles which are extended like this. Now these branchial aperture, they are guided by 4 lips. You can see 1, 2, 3 and 4. These are 4 muscular lips formed by the muscles, and even the atrial aperture, it is guided by 4 lips. Now in the branchial siphon and also the atrial siphon, the lower part, if we cut apart, if we remove the test, we can see that there are branchial tentacles at the base of this aperture, and these tentacles, they overlap each other to form a kind of sieve, so that the water entering in is filtered before it enters the body. The body is covered. You can see the internal structure of the body. The main cavity is this. This is the main cavity. Inside this is the main cavity, and it is covered by a leathery covering, and this is known as the test. So the body is covered by test, which is made by tunicin. The dorsal part of the animal is known as the body, and the ventral part is known as the foot. Actually, the body is represented by anterodorsal part. This is anterior and this is dorsal. Opposite to anterior, we say it's the posterior part. So the posterior part actually should be here, but it is not here. It is diagonally present to the anterior part, and this is the posterior part of the body. So when the antero-posterior axis is this, naturally the dorsal becomes this, the dorsal side becomes this one, and the ventral side is opposite to this and perpendicular to the antero-posterior axis, so the ventral part becomes this. So this is the strange symmetry that the animal is showing. The color of the animal is pink originally when it is born, when the animal is new, and as it ages and the test, it becomes opaque due to the deposition of many things which are present in the sea water. The animal is covered by the weeds and all, and different substances, they get stuck to it, and due to this it becomes translucent. In the beginning it is transparent, all the organs can be seen through the test, and later on when it ages it becomes translucent. The test. Now the test, as I've told you, is the outer covering of the animal, and it has many things embedded in it. These things are the test, you may say, is constituted of the matrix, like the general base of the layer, the matrix, in the matrix are embedded the cells. There are 7 different kinds of cells present in the matrix of Herdmania, and there are fibrils, and there are spicules, and there are vascular tubes. So this I've told you about shape and size, coloration, and test of the animal. And symmetry also I've already explained, antero-posterior and dorso-ventral. Let's come to the structure of test. As I've told you, there is a general matrix in which are lying the cells, the fibrils, the spicules, and the vascular tubes. Now let's take them one by one. First of all, let's explain what the matrix is. The matrix, it forms a general base of the test in which are embedded all these 4 things, and it is composed of tunicin. It is made up of tunicin. Now let's come to the cells. As listed, there are these kind of cells. First here, let us see the diagram. These diagrams, I did not give their heading because they are self-explanatory and there is no confusion about it. This is the kind of VS of the test, and in this you can see this dotted part is the matrix. Here it is labeled. This is the matrix of the test, and in these are present let's start from below. These are these spicule sheets containing the megascleres, These are the small spicules. This will come in the lecture afterwards. What types of spicules? These are megascleras and these are microscleras. And these are the test vessels. And these test vessels, they are giving out vascular ampullae on the surface of the skin, and obviously these vascular ampullae are tactile in function. Now we will do all these four structures in detail one by one. So let's come to the first type of the first cells which are embedded in the matrix, and these cells are large eosinophil cells. Now come to the lower diagram and you can see the types of cells listed over here. These are the large eosinophils. You can see they are the biggest than the other cells. They are the biggest cells as compared to all the other type of cells. So they are large, the size is big, and they are having a vesicular nucleus, vesicle-like nucleus in the center. The nucleus is also very big. Then come the small amoeboid cells. These cells, they are smaller than the large eosinophil cells, and their structure is amoeboid. They don't have a definite shape, and there is a clear nucleus present. Then come the small eosinophil. Why eosinophilus? Why are these cells called eosinophilus? Because they take up when they are stained, they take up the red eosin stain very well, and so they become red, and hence known as eosinophilus. The third type of cells are small eosinophilus cells. Their structure is same as large eosinophilus cells, only the size is small, and the other thing which is different is the nucleus. They don't have a vesicular nucleus, they have an eccentric nucleus. Eccentric means away from the center. So you can see the nucleus is not present in the center, it is lying on one side of the cell. And the structure, the size of the amoeboid cells, they are bigger than the smaller eosinophilous cells and smaller than the large eosinophilous cells. Then come the spherical vacuolated cells. These are cells which are having large number of vacuoles attached to each other, and their nucleus is not seen because it is hidden by these vacuoles. Then the granular receptor cells. They are oblong in structure and having a big well-defined nucleus, and they are sensory in function. Then the small branched nerve cells. These are the nerve cells and these are their branches. Then the last are the squamous epithelial cells. The squamous epithelial cells, they are hexagonal in shape. You must be knowing this already. And these kind of cells are also present in the matrix of Herdmania. After the cells, let's do the fibrils. Now the fibrils are also scattered in the matrix here and there, and these fibrils, they are exactly like the non-striated muscles of the chordates. You can also say that the fibrils are kind of structures which are these extensions of nerve cells. The nerve cells, they have these extensions. If we separate these from the nerve cell, these are kind of fibrils, and fibrils are present. These kind of fibrils are present in large number in the matrix of Herdmania. So, because the length of the lecture is quite big, so I am stopping over here, and we will continue next time. So thank you.